Hello, everyone. My name is Tracie Matsumoto. I'm a research horticulturist at the USDA-ARS Daniel K. Inouye Pacific Basin Agriculture Research Center in Hilo. And today I want to thank Andrea for inviting me to give a quick update on the evaluation of coffee varieties for Hawaii. And if you have any questions regarding the slides or presentation, feel free to give me an email, drop me an email at tracy.matsumoto@usda.gov. Okay, so today, I guess the main question that everyone asks when we start talking about varieties is when are the varieties going to be available to the growers? And so what I did today is kind of broke up the talk into some of the medium term goals in which we hope to get material to you maybe within the 1 or 2 years. This would include the World Coffee Research International Multi-Location Variety Trials, some coffee propagation of local varieties, as well as importation of varieties through a controlled import permit. For our longer term goals, I will talk a little bit about the coffee germplasm imports that we'll be bringing in from Cartier, as well as some of the breeding projects that we're collaborating to develop new varieties. So just as a reminder to everyone, so importation of coffee from international sources is prohibited to Hawaii or Puerto Rico. And so basically, to bring in anything from international sources, the coffee must be brought in through a controlled import permit through USDA APHIS. Similarly, with the Hawaii Department of Agriculture, importation of coffee plants or seeds for propagation requires a quarantine period, usually about 1 year. So any plants requires that you grow it under controlled conditions to prevent the introduction of pests and diseases to Hawaii. So just to back up and give you a little bit of background, um, we were fortunate to work with Dr. Chivumi Nagai, who's a plant breeder at the Hawaii Ag Research Center on Oahu. And so until October 2020, Hawaii was the only major coffee producing region without coffee leaf rust. Um, However, before the discovery of coffee rust in Hawaii, Dr. Nagai had actually started a breeding program for coffee leaf rust resistance in Hawaii. So she was able to make the crosses. However, because we did not have coffee leaf rust, there was no way for her to screen the populations to determine which ones are resistant to CLR. But she did build a valuable collaboration with Dr. Vitor Varzea in Portugal. And so it's that relationship that helped us actually identify coffee leaf rust when the race of coffee leaf rust when it was initially found in Hawaii. So we were able to send the spore samples from different parts of Hawaii to Dr. Varzea, and he was able to inoculate them onto the differential varieties and determine that looks like Hawaii had a single introduction of coffee leaf rust, and that turned out to be race 24. So in addition to breeding, some of or looking at breeding as a potential to develop new varieties in collaboration with Dr. Nagai in 2017. We also looked to work with World Coffee Research, and Hawaii became a participant in the International Multi-Location Variety Trial. So this slide shows you an overview of the World Coffee Research International Multi-Location Variety Trial, or we sometimes call it IMLVT for short. And so this slide just kind of summarizes all the countries that are participating in the study. If you notice, some of the countries have variety names under the name of the country, and so those are the countries that contributed their best varieties to test in locations around the world. And so out of the so 22 countries were participating in this study where they were able to take data on all these varieties to see how well they produce and how well they cup. And so Hawaii was fortunate to be actually the last participant in this variety trial. And so we did receive the last of the materials from this trial. And so I would like to direct you to the World Coffee Research webpage where you can actually find a lot of useful information on some that describes a lot of the varieties in the trial. You can search by the stature of the plant, whether it's a dwarf versus a tall plant, what the yield potential, optimal altitudes, as well as coffee leaf rust resistance, nematode resistance, or other diseases. And so you can get a lot of information on the varieties there. So, which varieties are in this trial? And so we kind of divided it up, the 30, into what is generally classified as susceptible versus tolerant to coffee leaf rust. But just as a caveat, or just to be clear, this is just general tolerance or resistance to coffee leaf rust and not necessarily specific to race 24, which is the race that we have in Hawaii. And so we are comparing all of these lines, but the one difference that we have from the rest of the trials is that we are comparing all of ours against Typica, since Typica is the industry standard for Hawaii. So this is just a cartoon, I guess, showing the setup of the variety trial. So they'll be kind of divided into tall varieties and dwarf varieties, and it'll be set up as a replicated Fisher block trial design. And so each block variety will be replicated 3 times in each block. So this is 30 varieties. And so with 30 plants, so it is going to be quite a large trial. We will be taking all the vegetative growth, CLR incidence, flowering data, cherry phenology, production data, bean quality, as well as cupping quality. And again, we are comparing this against Typica in locations including Kauai, Oahu, Maui, and Kona. And so we're also fortunate to have our nematologist, Dr. Roxana Myers, who will be screening these cultivars for root knot nematode resistance. So we would know if these eventually would need to be grafted or if they can be grown on their own root system once they get distributed to, hopefully get distributed to growers. And so just to make it clear, we are ARS is a part of this trial, and so we have a contract with World Coffee Research. However, unfortunately, this material is only available for research purposes. And so we are in the process of working with some of the breeders from the various countries to get the rights for Hawaii growers to grow these varieties and use them commercially. Okay. So, because we know we do have some IP issues with the material that is in the World Coffee Research IMLVT trial, and we wanted to find material that might be more readily available for growers. What we're also noticing is that some growers were noticing in their field to have green, healthy plants in a field that had been defoliated by coffee leaf rust. And so we're fortunate to work with our colleague, Dr. Daopeng Zhang, and ARS Beltsville in Maryland, and he has developed a small SNP panel for genotyping Arabica coffee. And so, just to give you a little background, a SNP is a single base change in a DNA sequence that occurs in a significant degree within a population. And what you would do is look at these little segments across the genome of a coffee plant, and from there you would be able to compare and show which group of coffee or what is the identity of that coffee plant. And so Dr. Zhang has been genotyping coffee from around the world, and he has also done 132 accessions from Cartier, which is the international coffee collection, and able to divide them up into 5 main groups, including your typical Bourbon group, East African varieties, Ethiopian land races, and wild germplasm, intergress varieties, and as well as the Canaflora or Robusta type of coffee. And so, with Dr. Zhang's help, what we were able to do is he was able to identify some of the material that we identified in the field. So just for an example, there's 3 called Juan, Cruz, and Maite. And so you can see that they all seem to have Ethiopia and Sarchamore in their background, with Maite also having a bit of Bourbon in the background as well. And so we hope to get the cupping of this variety hopefully within this growing season. But in the meantime, what we're doing is also working with our other collaborator at the University of Hawaii, Dr. Mike Shintaku, and his lab is funded by the USDA-ARS grant proposal, and they are doing mass propagation of coffee leaf rust resistant or tolerant lines. And so he's doing that through somatic embryogenesis. So hopefully by the time we get the cupping results, we will have plant material that might be available for growers to put into their field. Okay, so in addition to looking at what's existing here, we also wanted to pursue importation of coffee varieties through a controlled import permit. And so these are varieties that are available through one of our grower cooperators. These are not tied up in intellectual property, so these would be able to be grown in Hawaii without restrictions once we get it through the quarantine process. And so here are some of the varieties that we're currently trying to import through controlled import permits. Okay. You'll see that there's an Obata here, and so what we'll do is we'll compare that to the Obata that was already imported to Hawaii. So this slide I found that it was a nice summary of the different types of coffee that are available for commercial production, and shows you some of the names and their origins. And so you can see that the typical line does list Kona in it, and this is in that cyan color. In addition, you do see the Bourbons in pink, all of which are susceptible to coffee leaf rust. And so you also will see coming off of the Robusta branch in the light blue. And so you'll see the Timor hybrid, which Timor hybrid was the cross between the Caniflora and the Arabica. And so that one, you can see there's a Sachamore chain, a Sachamore line, as well as a Catamore line, and you can see the different varieties that have the Catamore in the background versus the Sachamore in the background. And so these are all coffee leaf rust resistant varieties. Okay, so I'm guessing that you're going to hear Dr. Mingli Wang from HARC will be covering that a bit more in her talk this morning as well. So looking at the longer term strategy, or I guess the 5-year plus. And so we were fortunate to have coffee added to our coffee plant germplasm system in 2019. And so because this was a new collection, we were able to kind of plan out how the material would be coming in. So working with our colleague, Dr. Zhang in Beltsville, he is genotyping the collections from around the world. We will be importing and putting them in quarantine in our facility at ARS in Hilo, where we are the primary collection. So we will have that in the field for evaluation. We do have a backup collection in Mayaguez. And so a core collection will be sent to Mayaguez for their analysis as well. And we're also working with scientists at ARS in Fort Collins to look at long-term preservation or cryopreservation of the material, germplasm material. And so working with Dr. Zhang, he was able to do coffee genotypes that are verified at Cartier, as well as some of the material that we're getting from HARC. And so you can see kind of the groups, how the populations or how the individual plants fit into different populations to avoid mislabeling. And so we'll know that our varieties will be true to type. We'll also be using that information to get the most diversity in our collection. This material will be imported to our Hawaii Gene Bank, and we'll be able to confirm these genotypes locally. So how is that information going to be used? So we're very, very fortunate to have our new research geneticist newly hired from at the end of last year. And so Dr. Chingyi Yu will be able to be working with Dr. Mingli Wang, who took over for Dr. Chivumi Nagai at HARC, as well as Dr. Lisa Keith, who's our plant pathologist here. And so I think Mingli probably talked about this, so I won't go into details, but basically it's just development of molecular markers to breed elite lines of CLR resistance into the elite Hawaiian coffee cultivars. Okay. In addition, what we're also going to be working on or working with is raw coffee research in the Innovia Global Arabica Breeding Network. And so this is to look more as a long-term strategy to, because we know that there's always going to be new coffee leaf rust races that will be resistant to the new varieties, so that we always want to hopefully, stay one step ahead. And so we'll be looking at new resistance genes to incorporate into our varieties so that we'll be ready for the next race, coffee leaf rust race, or other diseases coming into Hawaii. And so, in conclusion, I just want to thank our great collaborators, without whom the research could not get done. So it is truly a group effort to explore all the different avenues to bring these materials into Hawaii to hopefully give the Hawaii growers options in combating coffee leaf rust, as well as giving options to what type of coffee material they want to grow. I do want to thank our funding sources, which is the USDA NIFA SCRI, for that was given through SHAC. With Suzanne under Suzanne Schreiner, as well as HARC through Dr. Ming Li Wang. And I also want to especially thank our tissue culture technicians, Donna Ota and Elisa Mao, who are busily propagating these lines as we speak. And in addition, I want to thank the rest of the group in the plant unit, because I couldn't ask for a better group of people to work with. And so everything that we do is truly a team effort, and can't be done without everyone's support. And with that, I'll stop here and I guess join all of you later for questions. Thank you.